right, let's do it.
I'm Mark.
I'm John. We're working in a small optics lab at Darkling X with a prototype flow cytometer that has been designed for educational purposes. The system you see here is a fairly simple flow cytometer, and in the laboratory course sessions that we teach, we start out with the table essentially clear, and each of the modules that go into assembling this system is then sequentially put in place by the students, and over a time period of about 3.5 hours. At the end of that time period, they have an operating system that will look like what we're seeing here today. The critical parts of the system are flow chamber, where the sample is analyzed. The sample is fed from a syringe pump through this tube into the top of the flow chamber, and the sample is surrounded by a sheath fluid that comes from a pressurized tank back here with pressure control system. The sample is then sort of hydrodynamically focused into a middle of a stream and a measurement chamber, which is shown by this video projection. The sample, which should be on, but not very visible, shows up right in the middle here, and the laser beam comes in there. The sample is a fluorescent sample, and the fluorescence excited by a laser beam, which is a small laser pointer located over here on the left side. That laser beam is focused onto the flow chamber. The sample particles are excited by the focused laser beam. The fluorescence from the particles is collected by a lens in the back here and directed through a series of tubes and filters and another lens to focus it onto a photodetector in the back here, which is called a photomultiplier tube. The photomultiplier tube will generate an electrical signal that we then...
Which the signals come up into the preamplifier, and in the preamplifier they are amplified and can addition, and then they go out, and you can see the analog signals on the scope, and they also go into Azurite, which is the digital data acquisition system, where the analog signals are turned into numbers. They come out of Azurite via a USB cable and come into a computer running the program Cytometer, which when we start it, you can see the digital representation of the waveforms that are the same as the analog signals on the scope. You can also see a histogram of peak. The Cytometer is intuitive, which makes it easy to learn and easy to run.